I find my research really quite creative, actually, and what I like about science is that you get to pick exactly what you want to work on and what you find most interesting. Science can be quite creative, and synthetic biology in particular, to me, is really all about putting together components in new ways to create tools that will achieve something that hasn't been done before, or discovering new components, and so that you can use those new components in new ways. My research combines synthetic biology and molecular biology, and what I'm hoping to do is explore the natural diversity of proteins that nature has given us, particularly from viruses. The kind of virus we want to study is quite unusual. It's quite fascinating because it has a very special mechanism of replicating itself. And one is the class of viruses I want to study. They have what's known as a protein primed mode of DNA replication. And what you end up with is, at the end of their viral genome, some proteins called terminal proteins that are covalently linked onto the DNA. And that in particular is exceptionally rare in biology. There's very, very few examples of protein-DNA covalent linkages. And what I'd like to do is study that system. I think it has some really interesting properties. And after understanding it, then kind of exploiting it to create these new tools for gene editing. This is an area that's really important and quite hot at the moment, because there's hundreds of millions of people around the world living with and suffering from genetic diseases. So what we're looking for is some tools, some gene editing tools to correct and fix those mutated DNA sequences back to healthy sequences, and that would give a way to permanently cure patients from genetic disease. That's an application that I think studying these viruses will be particularly useful for. The research that we're going to be doing actually bridges across several different disciplines. First of all, we start off on the computational side of things, doing some bioinformatics, searching sequence databases, compiling sequences and doing sequence alignments and choosing which of these new sequences we'd like to study for the first time. Then secondly, we work in bacteria and we do a lot of DNA cloning, and we express these proteins inside E. coli, and we do protein expression and purification. The reason for that is then, thirdly, we move into more in vitro characterization, which is where we study proteins that we've just purified in a very simple and minimal system. That's a really nice way to understand their properties and their molecular details, having quite a controlled, stripped back environment where we're adding individual components in the test tube. And lastly, we want to leverage that new understanding for our applications, and that's where then we would move into mammalian cells, so human cells that we culture, or mouse cells, and try and do some gene editing experiments to actually change DNA sequence of these cells while they're still alive. It's an ambitious goal. It's going to take a lot of time and a lot of researchers' effort to make that happen. But along the way, there's going to be a lot of really interesting things that we pick up. So yes, we're going after this kind of interesting application for gene editing in the long run, but along the way, we're going to be studying really very understudied and rare class of virus, getting a lot of really fundamental biological insights for the first time, and just understanding these sequences that no one's really looked at before. I was an undergraduate here at Cambridge studying natural sciences, and I chose to specialize in chemistry. And the reason for that is that I've always really loved the problem-solving aspect of chemistry and how they answer and tackle problems. But actually, I always secretly found biology most interesting, topic-wise. I found nature and how things biologically work fascinating. And so what I was able to do was marry this problem-solving aspect of chemistry with the fascinating natural world of biology, and that's led me to really specialize. I did my PhD in molecular biology and synthetic biology, and I enjoy the ability to be able to combine solving problems, creating new tools, but doing that with biological components. I'm a new assistant professor here, and I'm just starting on my research program and looking to recruit new students and postdocs to help me out. the talent and expertise, the background of those scientists can be varied from more computational to more experimental. I think I'd like to give them a good training across the spectrum, so that they have an awareness of how their skills and skill sets fit in across like this whole program of research. But really, I'd like to take that opportunity to welcome people into the group. I do feel like a responsibility to mentor students well and train them, see them develop as people as well as scientists, and do some really interesting research together in whatever aspect they find most interesting to them. One thing I really like about science is that it's really challenging. By definition, you're doing something that hasn't been done before, or at least hoping to. You can get good at it, but it's difficult to master. And when you answer one question, often another question or two questions will pop up. And so as a career to get into, it's brilliant, because you know that you have this lifelong challenge ahead of you, something to grapple with, and that's very satisfying. In your approach as a scientist, a little bit of your personality comes out in how you ask questions, how you answer them experimentally, and I find that really quite satisfying and rewarding, that we get to do the work that we like in the way that we like to do it.